Hi students, this is Mr. Eckert, and we're going to go through Target H2, which says that you should be able to describe at least 3 different types of mutations in DNA and explain their effects on genes. So first of all, there's 2 main categories of any kind of mutations that we can discuss. One would be chromosomal mutations, which is not the focus of this target, but in a chromosomal mutation, there's changes in entire genes on a chromosome. So notice genes is plural, so that would affect multiple genes. So the focus on this target, rather, is going to be on gene mutations. Those are going to involve changes in just parts of genes. So in other words, individual nitrogen bases or letters that are in a gene. So in talking about gene mutations, there's 2 main types that you should be aware of: point mutations and frame shift mutations. So in a point mutation, and by the way, another name for a point mutation that you should be familiar with is a substitution mutation. So you should kind of add that to your notes in the margin. So point and substitution mutations are the same thing. That occurs when one base is switched out for another base. And you'll remember when you think about the structure of DNA that it has nitrogen bases that form the rungs of the DNA ladder, the pairs of nitrogen bases. And you'll remember that those are the bases adenine, thymine, cytosine, and guanine, so A, T, C, and G. So again, a point mutation is when one of those letters is switched out for another letter. Maybe the letter was supposed to be a T, and it switched to a G instead. Now remember what happens to the DNA during transcription. It's changed into messenger RNA, and that messenger RNA is going to leave the nucleus and be read by a ribosome, and you'll remember that it reads it 3 letters at a time, and every 3 letters, which are called codons, then correspond to amino acids that it's going to need to build the protein. So if we're only changing one base or one letter, that of course is only going to affect one amino acid. Excuse me. So that's a point mutation. Now a frame shift mutation is when one base is deleted from the DNA or added. So there's actually 2 kinds of frame shift mutations: frame shift deletion, frame shift addition. That's going to shift all the bases after it because remember, like we said, the ribosome, once it's reading the messenger RNA, it reads it 3 letters at a time. So if we're going to add a letter in or delete a letter, then that shifts how the ribosome reads that sequence. That's going to affect all the amino acids in that sequence after that mutation. So as we look at this example, that should make more sense to you. We've got a sentence here that you should copy in your notes. It says, biology students are really nice people. We're going to use this as a model of a gene where every word represents a codon. Now you remember that codons only consist of 3 letters at a time, and obviously there's most of the words, in fact all of them except for the word R, consist of more than 3 letters. But for modeling purposes, we're just going to pretend that this is a gene. Every word is a codon. Now we're going to do a point mutation. Remember, that's when one base is switched or substituted for another base. So we're going to substitute the R in the word R for a T. And now you can see the sentence reads, biology students ate really nice people. So we only changed one letter in one word. That only affected one word, one codon in a sense, but the whole meaning of the sentence was changed. And if this was a real gene coding for a particular protein, we would have changed one amino acid, and that could change the whole function, affect the whole function of that particular protein. So if you look at a codon chart, now you may not have one in front of you, but you could just open a new tab, go to Google, put in codon chart, and you should see, if you click on images, a whole bunch of codon charts. They're all the same. The codons code for the same amino acids in all living things. So you should see that there are multiple amino acids that are coded for by more than one codon. So because that's the case, you can see that you can have a mutation, a point mutation, actually change the codon, and just by random chance get another codon that codes for the same amino acid. So if you code for the same amino acid, you're of course going to get the same protein, because the amino acids are the ingredients for making the protein. So if you keep the same ingredient, you're still going to get the same protein. Those are called silent mutations because there's no visible effect then on the organism. Now in a frameshift addition, remember we're going to add a letter or a base, or you might think of it as repeating one. So what we're going to do is we're going to add an S after the word students. So again, remember, because the ribosome reads it codons at a time, it's going to shift everything after that addition. So now the sentence reads, biology students SAR ENIC equal E. And obviously that doesn't make any sense. So you can see every word or codon after the addition has been affected, and that is going to potentially change all those amino acids. So that's going to have a major effect on that particular protein. There's no way it's going to be functional. In a deletion mutation, which is just the opposite, of course we're going to delete a letter or a base from the sequence. So that S that made the word student plural, we're going to take it out. Everything shifts to the left, and now the sentence reads biology studenta ren ICEP EPOL. Again, all the codons have been changed after the mutation. That's going to change the amino acids, and obviously the protein is not going to be functional, just like that sentence is nonsensical. So how do these different mutations affect organisms? Well, it can be anything from a silent mutation, which has absolutely no effect at all. It can be a relatively minor effect, like if, let's say this was a protein that controls whether or not you can roll your tongue. In that case, a mutation in that, if you're no longer able to roll your tongue, that would be, I'm sure you'd agree, a minor effect. It's not going to really affect the quality of your life. It can be severe. It can be lethal, as you saw if we watched the Tay-Sachs video clip in class. That's a lethal point mutation, only changing one letter. Or they can actually be beneficial. Many people have the misconception that all mutations are harmful, but they're not. In fact, beneficial mutations are what drives evolution. And we'll talk about that more, of course, in the evolution unit, but there are beneficial mutations. So what you really want to think about, though, is the protein. How has the protein been affected? Because proteins are made of amino acids, those are the ingredients. If we start changing those ingredients, we're obviously going to change the protein, and the protein is not going to function the way it was supposed to. And again, here's a reference to that Tay-Sachs video clip. If we have not seen this in class, you might want to look it up on YouTube. If you just type in one wrong letter and then Tay-Sachs, you should see a video clip of an individual, a little boy who ended up with Tay-Sachs and the effects of it. So that does it for this target. If you have any questions, please make sure you ask in class.